Hey there. Have you ever just looked at something and wondered, what is this really made of? From the water we drink to the salt on our table, how much of it is actually the one pure thing we think it is? Well, today we're going to find out.
Okay, take a look at this. It's Himalayan rock salt. It's beautiful, right? We see it everywhere, in fancy kitchens, on restaurant menus. It just looks so natural and pure with that amazing pink color that we're told comes from all its healthy minerals. But here's the question that really gets to the heart of what we're talking about today. From a strict scientific point of view, is this salt actually pure? I mean, could the very thing that gives it that famous color be telling us a completely different story? All right, so here's how we're going to break this all down. First, we'll get on the same page about what pure really means to a scientist. Then, we'll dive into how things mix together to make solutions. After that, we'll look at solubility. Yep, the breaking point of a mixture. And finally, we'll uncover the super clever way science can take it all apart again.
So let's get right into it. What does pure really mean? Because, in everyday life, pure might mean something is natural or clean or organic, but in the world of chemistry, it has a much, much more specific meaning.
And this is it. It's actually pretty simple. A pure substance is made of only one type of thing, either atoms of just one element, like a bar of pure gold, or molecules of just one compound, like perfectly pure water, H2O. There's nothing else mixed in. That's it. It's a very strict definition. And because that definition is so strict, it leads to a pretty surprising truth. Almost nothing you run into in your daily life is perfectly pure. So that Himalayan salt, its famous pink color comes from traces of other minerals, like iron oxide. So scientifically speaking, it's not pure salt at all. It's a mixture. So that begs a pretty important question, right? If we can't always trust what we see, how do scientists actually check for purity? Is there some kind of definitive test to know for sure? Turns out there is. You can think of it like a physical fingerprint. Every single pure substance melts and boils at a very specific, unchanging temperature. We all know pure water boils at 100 degrees Celsius at sea level, always. But the second you add impurities, like salt, that temperature changes. It's a dead giveaway that you're dealing with a mixture.
Okay, so now we know that most things around us are actually mixtures. So let's move on and figure out how these mixtures get made in the first place. Welcome to the world of solutions.
Now, to really get a handle on solutions, we just need to know the three main players. You've got the solvent, which is the liquid doing all the work, think water. Then you have the solute, which is the stuff that gets dissolved, like sugar. And when you mix them together, bam, you get the final product, a solution. And dissolving isn't some kind of magic trick where the solute just vanishes. As this little model shows, the particles of the solute, those little green things, are simply breaking apart and slotting themselves into the empty spaces between the solvent particles. They're all still there, just spread out and mingling.
This shows the idea of concentration perfectly. See the beaker on the left? Just a few solute particles floating around. That's a low concentration, or what you might call a weak solution. Now the beaker on the right is totally packed with them. That's a high concentration. It's really just about how much stuff is crammed into the same amount of liquid.
So what happens if you just keep adding more and more solute? You've been there, right? Stirring sugar into iced tea, and eventually you hit a point where it just won't dissolve anymore. It just sinks to the bottom. Well, that brings us to our next topic, solubility, the breaking point.
This limit is what scientists call solubility. It's the absolute maximum amount of a solute that you can possibly dissolve in a solvent, but, and this is key, at a specific temperature. Once you hit that point, the solution is called saturated. It's full. It literally cannot take any more.
Now, this is where things get really, really interesting, because temperature can totally change the game. Just look at this graph. The red line for potassium nitrate, wow. As the temperature goes up, its solubility just skyrockets. You can dissolve way more of it. But then look at the blue line for sodium chloride. Regular table salt. Heating it up barely makes a difference at all.
So why does heat usually help things dissolve? Well, it all comes down to energy. When you heat up a solution, all those tiny particles, both the solvent and the solute, they start vibrating and sipping around like crazy. All that extra energy and movement helps them break apart from each other and mix together much more easily.
Okay, we've figured out how to spot purity and how to make mixtures. So for our last section, we get to the really clever part, science's separating secret.
How do chemists take a complicated mixture and unmix it back into its pure, individual parts? Well, one of the most elegant ways is a technique called paper chromatography. It sounds fancy, but it's a beautifully simple three-step process. First, you put a spot of your mixture, say black ink, onto a piece of special paper. Second, you just dip the edge of that paper into a solvent. And third, you watch the magic as the solvent soaks up the paper, separating that single black dot into all of its different colors.
The best way to think about it is like a race. As the solvent moves up the paper, it carries all the different substances in the ink along for the ride. But some substances are more soluble. They dissolve more easily, so they travel farther and faster up the paper. The less soluble ones, they get left behind.
Now, this might just seem like a cool science fair experiment. And don't get me wrong, it is. But it is so much more than that. Which brings us to the really crucial question. Why is this simple separation technique so incredibly important in the real world?
Well, for one thing, it helps keep our food safe. This picture shows a more advanced version called column chromatography. Food scientists use this to check for things like unwanted additives or to see if a product has started to spoil, because they can detect the specific chemicals that are produced during decay.
And honestly, the applications are just everywhere. Forensic scientists use it to analyze tiny samples of ink from a ransom note or blood from a crime scene. It's used in medicine to help purify and prepare life-saving vaccines. Environmental agencies use it to detect dangerous pollutants or pesticides in our water supply. It's a true workhorse of modern science.
So, we've gone all the way from a simple grain of salt to a powerful technique that helps solve crimes and save lives. And it all comes down to understanding what things are really made of. It kind of makes you wonder, right? Now that we have these incredible ways to separate even the most complex mixtures, what hidden secrets hiding inside the materials all around us are we going to discover next?